Welcome, everyone, to the Roger E. Kepi Endowed Lectureship Series 2020. I think this is one of the first lectures this department has ever given on Zoom or on the internet. And so it's quite an exciting time for all of us. I wish it was due to different circumstances, and I certainly wish that our guest speaker, Celia Schiffer, was here so we could all meet her and go out to dinner and hear the wonderful things that she does. But COVID-19 stepped in, and here we are, everyone. But that doesn't mean we still can't have fun and enjoy each other's company, and more importantly, get a good taste of some fantastic science coming out of Professor Schiffer's laboratory. And so I know that's going to excite all of us. Before I introduce the speaker, though, I'd like to talk a little bit about Dr. Roger E. Kepi. He passed away in 2009, not so long ago, and his wife followed in 2012. Roger joined the Department of Biochemistry at Oklahoma State University in 1959 as an associate professor, and in 1963 was promoted to full professor. He served as the fourth head of the department for an unprecedented 28 years, from 1963 until his retirement in 1990. Once again, I don't know many people who stay as a department head for 28 years. In fact, he's the only one that I'm aware of. Most people stay 5 to 10 years and then realize how difficult it is and they move on. Soon after his retirement, Roger provided a generous endowment to establish the Roger E. Kepi Endowed Lectureship Series, known to our faculty and students as the Rekel Seminar. So this is our quintessential seminar of the entire year. And we only invite absolutely the best scientists that we can find out there. Over the years, the Rekel Seminar was the highlight of the Departmental Summer Symposium, bringing a host of esteemed scientists to the OSU campus. Prior to Roger's death in 2009, the Kepi Endowed Lecture was incorporated into our departmental seminar series with talks that reflect Roger's interest in amino acid metabolism. That since has changed, and we've moved on to numerous different subject matters. The donors have been happy with the speakers that we've been bringing in for this lecture series. This change coincided with the naming of the Roger E. Kepi Seminar Room to honor the years of service provided by our long-term department head and the establishment of the Roger E. and Norma L. Kepi Biochemistry Scholarship. So in our department, we always say, give like a Kepi. And they've been wonderful in terms of providing a huge number of opportunities, both for us as faculty and also in students and staff, to witness some fantastic science going on and having fantastic speakers. But more so, they provide money for our students, you know, our bread and butter of every university. And the reason why I love being a department head and a faculty member, because I get to interact with those students. It just brings me great joy that the Kepis thought long-term in terms of being able to provide those people with opportunities and financial assistance so that they can continue being students and not being worried about those kind of things. All right, so with a big thank you to Roger E. Kepi, round of applause for Roger. I'd like to move on to introducing the speaker. And what a speaker we have today. I have some notes here. Professor Celia Schiffer is a professor in the Department of Biochemistry and Molecular Pharmacology. She's also the Gladys Martin Smith Chair in Oncology. Celia, if I get something wrong, please speak up, by all means. I don't want to get this wrong. And she's also the founder and director of the Institute for Drug Resistance at the University of Massachusetts Medical School, an incredible institution. Dr. Schiffer has a BA in physics from the University of Chicago, and a PhD in biophysics from the University of California, San Francisco. And she did some postdoctoral training at the ETH, which is the national institution of Switzerland in Zurich. And she also worked at Genentech while she was there. I presume that was also for a postdoctoral experience. Yeah. What a wonderful opportunity. And not only that, during that time, you're really on the ground floor in terms of the technologies they were pursuing to produce products. Wow, that's fantastic. She has directed numerous collaborative NIH and other grants totaling over $45 million to date, which is just an exceptional amount of grants personship that I'm just completely moved by that number as a department head. Including 3 NIH Program Project Awards from the National Institute of General Medical Sciences. She has published over 170 peer-reviewed journal articles, and I took a look at Google Scholar, as I often find myself doing as an administrator trying to support faculty. She has a total of 8,154 citations as of today, and she has an H index of 53, and I think a median index for people who want to become a National Academy of Sciences member is 54. So you're only one off, Celia, so you'll get there eventually, that's for certain.
Thank you.
Go on. Okay. And so 170 peer-reviewed journal articles using a combination of experimental and computational structural biology and chemistry techniques to obtain key insights into drug resistance. Okay. She was also was part of the leadership that started Mass Theory, and I couldn't find what that was. Celia, can you describe what Mass Theory is?
It was an entrepreneurship organization at our university to sort of help facilitate entrepreneurship of the faculty.
Oh, fantastic. You know, Steve Hartson, one of our faculty members here, he taught me a term, applied horizon. And so now when I hire assistant professors, I always say, you know, you should have at least a 5 to 10% applied horizon. Keep your idea on potential products or technology that you produce that you can go out and make some money for yourself and the institution. So I love that idea. And also she had a major leadership role in the regional cryo-EM facility at the University of Massachusetts Medical School as well. And of course, we're all salivating about that because of the fact that you have one and we don't. And so that's quite exciting. In 2015, she became a fellow of the American Academy of Microbiology. In 2010, she received the UMass Medical School's Outstanding Mentoring to Women Faculty Award, which is so incredibly important in terms of advancing more women, in particular in the STEM disciplines. So good on you there, Professor Schiffer. In 2016, she was honored by the inaugural Chancellor's Award for Excellence in Mentoring. And in October 2016, she was awarded the Massachusetts Society for Medical Research Educator of the Year. So not only are we talking about an incredible researcher, but also an incredible educator. And getting science out to the public, as we all know, is incredibly important, especially moving forward in this present pandemic that we exist in. I also noticed that the American Society for Biochemistry and Molecular Biology awarded her the Rose Award in 2020. Just an exceptional award to recognize all she's done during her career. So Dr. Schiffer has developed a strategy to lessen the probability that drug resistance occurs. She has done so at the initial stage of drug development when the drug is first being designed. She realized that through understanding the molecular mechanism by which the disease process occurs, she could develop inhibitors that block the disease in such a way that the likelihood of resistance occurring is greatly reduced. Well, I just love that idea, and I'd love to see it applied to all microbes, and I have a feeling we're going to hear about that today. She demonstrated this strategy with targets in 2 globally pandemic viruses, HIV and hepatitis C, and is showing this strategy generalizable to many other diseases. By designing drugs to be less susceptible to drug resistance, the drugs are more likely to last and stay effective. And of course, that's critical. Antimicrobial resistance has not gone away, and the battle is still being waged. Finding other general parallels to avoid drug resistance in drug design and patient therapy motivated her to conceptualize and co-found the Institute for Drug Resistance, as drugs that retain their ability to be effective is ultimately better for both the individual patient and for society as a whole. So big round of applause for Professor Schiffer for taking her time to come and give us a lecture. And once again, I couldn't think of a better person to pick for the Rekel lectureship. So thank you, Celia, so much for giving us the opportunity to see your research. And with that, I'm going to let you take it over.
All right. Thank you so much, Professor Gustafson. It's really an honor for me. That was an amazing introduction, and I don't know, have to live up to that introduction, but it's really a special honor to be able to present the Renek lecture this year, and it sounds like Professor Kepi was an amazing mentor and colleague and visionary supporting science in your department and your university, and I'm really grateful that he has shown such leadership, and sounds like it would have been an amazing person to meet and know. Um, so you guys are, I assume, can see my slides, and everything looks okay?
Looks great, Celia, and I can hear you fine.
Okay. All right. So let me just. All right. So today I'm going to be telling you about uh, combating drug resistance. And I'm going to tell you about a series of different stories from my laboratory, basically from base editing to machine learning uh, in drug design. And feel free, if you have questions as I go, to interrupt me. Just say my name. I won't be able to see if you put something in the chat box while I'm giving the lecture, but I'm happy to discuss, answer questions as I go, or at the end, as you like. So, as all of you know, drug resistance is a huge problem in our society. It is impacting millions of people and costing billions of dollars every year. And basically, it occurs anytime we have a quickly evolving disease or system. So, I'm going to talk today about antivirals, HIV, hepatitis C, and so forth. And especially in this age when I can't be there in person because of this SARS-CoV-2. I mean, we all understand the importance of microbes, but resistance occurs not only in viruses, but it occurs in bacteria and in oncology and cancer. All of these areas are where things are evolving quickly, and we try to restrict their growth. Resistance occurs, and it also extends beyond our human diseases to things like herbicides and pesticides, where resistance can also occur pretty quickly. So how can we think about avoiding resistance? Can we design drugs that are less susceptible to resistance? Basically, drug resistance can occur anytime there is rapid evolution in such a way that what we're targeting is able to maintain function while no longer being inhibited by the drug. And in today's talk, I'm going to talk about when the target evolves resistance. There are other mechanisms of resistance, but I'm going to focus on the evolution of target resistance. A lot of the ways that we do modern drug design fail fail to avoid resistance because they don't pay attention enough to function. So whether we're doing high throughput screening or structure-based drug design, that we're leaving opportunities for resistance to occur that don't compromise function. And I'll explain more in detail what I mean by this in a moment. The sort of take-home message is, though, that disrupting a drug target's activity is necessary, but not sufficient for developing a robust drug that avoids resistance. We really want to be able to design drugs to last, and not just at the moment, but so that we can avoid the most frequent evolutionary things that are happening. I mean, we're all working. This is an amazing time in biology. I mean, the levels of data that we get, whether it's in the fields of structural biology and chemistry that I work in, or thinking about sequencing and in the microbiome and metabolomics and proteomics, the trick with all this data and at all these different levels is, how do we integrate it? How do we integrate many different strategies to be able to understand biological processes or events? And I think the more we understand this bringing together of different levels of science, the more we'll be able to transform our understanding of biology. In my laboratory, we try to integrate a very multidisciplinary approach. We're structural biologists largely and have solved many different crystal structures, some of which I'll tell you about today. But we also do a lot of organic synthesis, a lot of computational work with molecular dynamics and machine learning. We think a lot about the sequences of the targets from patient samples as well as from viral culture and understanding what it is. And all of these things we bring together through collaborations, virology and NMR. We bring together in a new drug design strategy where we try to avoid drug resistance by leveraging evolutionarily constrained places and realizing before any drug is given that often there exists a heterogeneous population of viruses or cancer cells or weeds for that matter that are able to accommodate the selective pressure of the drug or the inhibitor and develop drug resistance. So it's the adaptation occurs throughout this process, and it may not be only restricted to the drug target. It's really adaptation of the entire system that needs to be considered. So with this, we're trying to develop a new strategy. This is my current lab. So I'm lead a diverse array of students, postdocs, and research faculty in our efforts. And together we're more than the sum of our parts, so we can solve these complex problems. And we look at many different systems, a lot of work on viral proteases, which I'll speak about quite a bit today, but also different aspects of influenza, doing some cryo-EM, and I'm also going to touch on some work we're doing with the SARS-CoV-2 that we've initiated in light of the current pandemic, and research on a set of enzymes that are cytidine deaminases, the ApoBec3s, where we're trying to understand how diversity gets entered into a system. So how do we avoid drug resistance? As a structural biologist, our perspective is really looking at the interface between evolution and structure-based drug design. So drug resistance often occurs where mutations occur in the drug target that allow it to retain the function while no longer being efficiently inhibited by the drug. And so if you think about a drug target, most of our drug targets are enzymes, and these enzymes work on particular substrates, and that forms a complex of the substrate. It has its course of action, product is released, but if it's on a disease pathway, the disease progresses and you have an unhappy patient. So if we think about how we do drug design against the enzyme target, the way we usually do so is we screen against a particular target, either a high throughput screening or structure-based drug design screening. And we screen against a target, a bunch of small molecules against the target. And if, so I see there's a chat window. If somebody needs my attention, just feel free to say something because I can't actually read the chat. So we screen against a target, and if we're lucky, we can find a hit that blocks the enzyme target from functioning. And if by blocking this from functioning, if we're really lucky, it's bioavailable, it's a good compound, lead optimization to make a good drug. I mean, a good drug has to have many different properties to work properly. And once in a quickly evolving disease, however, this can become problematic because in a quickly evolving disease, this enzyme target is not going to stay the same and can evolve drug resistance. And once resistance occurs, the inhibitor won't bind. So this resistance can occur by a number of mechanisms. It can occur because of infidelity of the reverse transcriptase or polymerase of a virus. It can also occur because of base editing properties of enzymes like ApoBec3s or other base editing enzymes. And so once those resistance occurs, the inhibitor doesn't bind so well anymore, but the enzyme's able to continue to recognize and process its substrates. So our new paradigm in drug resistance is to really pay attention to the enzyme substrate complex and use that as an added constraint in structure-based drug design. So we look at if the enzyme interacts with more than one substrate, so much the better, because then it is more evolutionarily constrained. And we design inhibitors to fit within what we call the substrate envelopes. And this substrate, these inhibitors should be still be potent, still should be bioavailable, and all the other good things an inhibitor must be. But they, by fitting within the substrate envelope, they should be less susceptible to resistance, because a mutation impacting these inhibitors will not only impact the binding of the inhibitor, but will simultaneously impact the recognition and processing of the substrate. So an inhibitor, so by doing so, the inhibitor is more robust against resistance, and the inhibitor should maintain their potency against pre-existing resistance or other polymorphisms that might exist in the enzyme target. So if we compare our method to others, you can see that traditional drug design, be it structure-based or high-throughput screening, there's many opportunities for resistance to occur. By staying within the substrate envelope, we're trying to decrease the probability of resistance occurring, with the idea, again, that disrupting a drug target's activity is necessary but not sufficient for developing a robust drug that avoids resistance. Another way of thinking about this is that we're looking at how resistance occurs when the biological function of the target is favored over its inhibition. So you can think of this as a balancing act, and this balancing act of potency can be affected by changes in the environment. It can be also be affected by changes in the structure or the dynamics of the free or bound state of the target, or the flexibility of the inhibitor. I mean, going back to your freshman chemistry class, that delta G equals delta H minus T delta S, entropy and enthalpy are key and critical in understanding how resistance occurs. So it's not just the structure of a complex, but it's really the dynamics and flexibility by which the inhibitor fits to the target that becomes very important in understanding the molecular mechanisms for resistance. So the way we've been thinking about this recently is that drug resistance is really a multidimensional landscape. And we're trying to parse this multi-dimensional landscape. So we think about the protein structure and its dynamics and its flexibility. We also think about the drug structure and its chemical makeup and its potency to the drug target. But on a third or yet another axis, if you like, we think about the sequence diversity of the target and how that sequence diversity might vary between different viral genomes, between different cancer genomes, and so forth. So we're really parsing this multi-dimensional landscape when we're doing inhibitor design to avoid resistance, to find sort of the low energy position where an inhibitor might bind and avoid resistance. So in today's talk, I'm going to give you a series of sort of 3.5 vignettes. I'm going to tell you about, and as I said earlier, feel free to interrupt me whenever if you have questions. I'm going to tell you initially about thinking about ApoBec3s, and these are these base editors that are involved in our innate immune system and we suspect contribute quite a bit to resistance, especially in oncology. I'll tell you about some drug design efforts we've done on hepatitis C, where we've really demonstrated how the substrate envelope can work in avoiding resistance, and have some very promising data on the hepatitis C protease. I'm going to briefly talk about translating some of these concepts to the SARS-CoV-2. Uh, Given the current pandemic, global pandemic, my lab, as well as many other labs, has shifted gears to try to address some of these problems and see if we can shed light on, have some insights in this area as well. And finally, I'm going to end on some of our efforts using machine learning to decipher how remote mutations confer resistance to an enzyme active site. So first story is about these base editors, the ApoBec3s, and how our own innate immune system might contribute to resistance. The ApoBec3s are cytidine deaminases, and they basically deaminate single-stranded DNA or RNA and introduce C to U mutations. Their signatures are coming up more and more in cancers of all sorts, from breast to bladder to lung cancer. And by having diversity in the genome, this is essentially facilitating resistance. It's fascinating that ApoBec3s are only in placental mammals, and they actually are very diverse even among placental mammals, with mice only having a single ApoBec3, where humans have 7 different ApoBec3s on the same chromosome. These ApoBecs are actually some of the fastest evolving proteins in the human genome. And even if we look at one of the 7 ApoBec3s in humans, ApoBec3H, there are 7 different haplotypes in humans. So if your ancestors came from different parts of the world, the people on this call, we probably have quite different ApoBec3s just serving the people on the call. So these enzymes have been evolving quickly, and it's been hypothesized that maybe this is part of their role in the innate immune system, and being able to respond to different viruses or different infections in different parts of the world. But that still remains to be seen. ApoBec3s enzymes appear to have many different complex roles, and a lot of this is still being sorted out. I mean, this CDU mutation, as I already mentioned, has shown up in a lot of different cancers, but the ApoBec3s were initially discovered in restricting HIV, and have been shown to restrict other viruses. HIV actually has a particular protein that targets ApoBec3s for proteasomal degradation. But the AID enzyme actually is involved with adaptive immunity and changing how our antibodies respond to things. And they've been implicated in double-strand break repair and in retrotransposon restrictions. So there's all sorts of roles, and I think there's a lot more that we'll learn in future years about these enzymes. But they're clearly very powerful and important proteins, because they keep evolving within the even human beings around the world. They've also been in the news lately, and maybe some of you have seen, there have been a series of papers, the first one in 2016, where these ApoBec3s that are the base editors that are being connected to various CRISPRs. So they add much more specificity in ability to do base editing than CRISPR alone. So David Liu and his colleagues have initiated a lot of that work, and Beam Therapeutics has been doing very well with this idea of adding ApoBecs to CRISPR-9, Cas9, and other CRISPR enzymes. But why are there so many apobecs? What's sort of, why do we need so many? And if we look at these enzymes structurally, there are four loops around the active site that are very different in their amino acid sequence and must be responsible for the differential specificity. These loops are all beyond the central structural topology of the enzyme. But as you can see in gray, there's a lot of sequence variation between these four loops. The conservation is only sort of back in the active site and at the base of the loops when you're in beta strands. But these loops are what determines the differential specificity of the different apobecs. And I think, we're still don't completely understand the substrate specificity of these enzymes. We're beginning to understand, but I think there's a lot of structural insights that are going to inform the specificities beyond what has been published on so far by us and other laboratories. My lab has been working on these enzymes for over the last decade or more, and we've solved a series of structures of different forms of these enzymes. Some of them in my own lab and in collaboration with Dr. Hiroshi Matsuyo at Leidos at NCI Frederick, and we've solved these structures, both the apo forms and in complex with single-stranded DNA. And so we're beginning to understand the molecular basis for the specificity of these varied enzymes. So if we look at, this is a structure that we solved a few years ago of apobec 3A and in complex with single-stranded DNA. And you can see how the single-stranded DNA wraps around what this is a histidine 29, which is sort of a gatekeeper that locks the DNA into the active site. And for deamination, and once the CDU mutation occurs, then that pushes the histidine back out and allows the single-stranded DNA to escape. But this complex is stabilized by a whole network of hydrogen bonds and ionic interactions that go around the DNA and are allow the enzyme to recognize this specific sequence. So to figure out and understand the specificity even better, we've used molecular dynamics simulations. Molecular dynamics simulations is a computational way of solving Newton's equations of motion to look at the dynamics of a system. And often by doing what we call parallel molecular dynamics, we look at subtly different systems to try to get different aspects. And it's basically using classical mechanics considerations to understand how the system works. So when performing molecular dynamics on this simulation, my recent PhD student, Sharon Kuo, here's a molecular dynamics simulation. You can see that the single-stranded DNA is very stable in the active site here, with hydrogen bonds stabilizing it into the active site. So we're pretty confident that the crystal structure in the conformation of the single-stranded DNA is pretty good, and the histidine stays put, and everything looks very stable. And so we take this information and take it a step further and say, hey, can we design, sorry, oh, I have to click a different side of the, so using methods of molecular dynamics, we look at complexes of single-stranded DNA with A3A, A3B, and A3G, three different forms of this enzyme, and can understand the differential specificity, both by looking at the crystal structures and molecular models and molecular dynamic simulations of these different different types of apobec 3s that exist in humans. So you can see that how A3A binds is somewhat different than how A3B or A3G binds, where it's much more extended. So those four loops that I pointed out a couple slides ago really distinguish the differential specificity. By looking at this structure, so this is now A3G and crystal structure that our colleague Hiroshi Matsuya's lab solved, we can really understand how the DNA fits within the active site. And by paying attention to all the hydrogen bonds and all the detailed interactions, we can start to think about how could we inhibit these enzymes, and maybe by inhibiting these enzymes, we can decrease the diversity that occurs perhaps in cancer. And so if you think about it, you know, the C changes to a U with the enzymatic activity of the enzyme, and that we could uh, change the base to some sort of modified base by changing the particular sequences that exist, like in this crystal structure. And from the crystal structure, we can discern uh, whether the conformation of the DNA, whether it's uh, you know, C3 and C3 prime endo or C2 prime endo, whether the puckering of the sugar, whether they're syn or anti. And we are working now, a current student in my group, Adam Hedges, is working with a colleague of mine, Jonathan Watts, to say, can we modify the bases and develop inhibitors by modifying the nucleic acid. So both at the base level and in terms of the sugar conformations. We can do this both by replacing the bases by analogs like this one that's been used before, zibularine as a cytidine deaminase inhibitor, or by modifying the DNA to stabilize its conformation, as Jonathan Watts has done previously. So, I mean, this is a story that, you know, we're just beginning to do. We tried, Adam synthesized the single-stranded DNA with a zibularine in the catalytic site. And by doing so, he was able to test, I mean, other people had done this, and Shrung modeled it based on our crystal structure, but he went through the synthetic pathway, incorporated the zibularine, and was able to show that there was some inhibition with this modified single-stranded DNA. And this Ki is in, although still very weak, nowhere near potent enough to be of clinical interest. It's at least a good initial start, and it's in line with what others have observed for incorporation of zibularine within single-stranded DNA. So, I mean, this is sort of our first step of inhibiting ApoBec3s. And, you know, hopefully inhibiting ApoBec3s should decrease the diversity within cancer cells, and thereby, by decreasing diversity, decrease the chance of resistance. So you can imagine, you know, maybe if you had an inhibitor to ApoBec3, you could combine it in making a new drugs in oncology, possibly in combination with other drugs, a combination therapy where you might have a kinase inhibitor, but then simultaneously have an ApoBec3 inhibitor to decrease the diversity and hopefully thus potentiate the longevity of the kinase inhibitor in treating the patient. But you'll stay tuned. Hopefully there's more to come in the coming years. Okay, so now I'm going to switch gears and tell you about the second story. The second story is working on the hepatitis C protease inhibitors, and hepatitis C protease is a major drug target in the treatment of hepatitis C. I think the numbers I heard is something like 70 million people are infected with hepatitis C. And hepatitis C, though, luckily has come a long way. Hepatitis C is now a curable disease. A decade ago, it wasn't, but now through combination therapy, many using a hepatitis C protease inhibitor were able to cure most people infected with hepatitis C. I mean, hepatitis C is a tricky disease because people often get infected with it and they don't know for decades till decades later that they're infected when their liver starts to cirrhose. So for a long time, all doctors could do was to do liver transplants. But now, when they find out that somebody's infected, usually a combination therapy with a protease inhibitor uh, uh, cures patients of these diseases. And here's the three most recently approved hepatitis C protease inhibitors that are very potent, and often people can be cured in as little as 6 weeks. So that's pretty amazing. But these are compounds from major drug companies. So hepatitis C is a serine protease. It is responsible for cutting the viral polyprotein in a number of different sites and allowing viral maturation. So inhibiting this protease is a good way of stopping viral maturation and thus the infection. Like I told you in the beginning of the talk, we think about things in terms of substrate envelopes. And with hepatitis C protease, it recognizes a whole series of different substrate sequences that have conserved homology at the P1 and P1 prime, and interestingly at the P6 position, but have quite diverse sequences elsewhere in the sequence. A while back, we solved a series of crystal structures of substrate complexes. We were able to calculate a substrate envelope, sort of like I showed you before in this cartoon, where the consensus volume of the various substrates were fitting. So we were what the enzyme seems to recognize is this conserved three-dimensional shape rather than an exact amino acid sequence. Drug resistance comes up in hepatitis C pretty readily. There are three primary sites of resistance, and they occur right outside the substrate envelope, right outside the catalytic triad, sort of like I showed you in the cartoon before. The sites of resistance are primarily this arginine that goes to a lysine, this little alanine here that goes to valine or threonine, or this aspartic acid that goes to an alanine. The way the resistance occurs similar to how I showed you in the cartoon. So you can see at this P2 site of the inhibitor is protruding out of the substrate envelope and making an aromatic stacking on the arginine here. So if you can imagine, if the arginine mutates to lysine can no longer make this aromatic stacking and you get resistance. Or if this aspartic acid goes to a small alanine, you lose this salt bridge between these two arginines, and again you get resistance. This inhibitor can tolerate changes at this small alanine getting slightly larger because it has a little bit of flexibility between these two moieties. I also want to draw your attention to this inhibitor has a macrocycle, and that macrocycle is called a P1, P3 macrocycle, and it's fitting pretty well within the substrate envelope in the background. So let me see if I can get the movie to stop. There we go. This is very much like the cartoon I showed you with the inhibitor protruding out, and where it protrudes out is where the resistance occurs. But this inhibitor has some good points in that it has this macrocycle that fits within the envelope that you see back here. Another inhibitor that is actually, so tenofovir, this inhibitor was in clinical trials but never actually made it to the clinic. Another inhibitor that we looked at is grisaprevir, and this is a compound that is in the clinic, is made by Merck. And when we solved the crystal structure of this, we found that it also protrudes from the substrate envelope, but it does so in an interesting way. Rather than packing on this arginine like the previous inhibitor, it's packing on the catalytic triad. So although it's sticking out from the substrate envelope, by packing on the catalytic triad, it's a little bit more resistance proof because the enzyme can't tolerate the catalytic triad mutating without the enzymatic function being completely compromised. However, this inhibitor has a very rigid macrocycle here. And while it's not so susceptible to mutations at this arginine, it is susceptible to changes at this little alanine going to a valine or threonine because it pops it right out of the active site. And it's also susceptible to this aspartic acid going to an alanine because you lose the packing of the macrocycle. So so although this inhibitor, grazoprevir, has some good parts, there are also, of packing on the catalytic triad, there are also some problems with the rigid macrocycle making it susceptible to resistance. So the trick for us was really to see, can we combine the best characteristics of both inhibitors to do better. And actually, before I get to that, I mean, keep in mind this grazoprevir, this macrocycle, this rigid macrocycle. Well, let me turn back for a moment to the inhibitors that are currently used in the clinic. Here's grazoprevir, and here are the two other compounds made by Gilead and Abbott. And I'm not an organic chemist. I work with Dr. Akbar Ali, who's a very talented organic chemist. But, you know, to all of our eyes, I hope you can see these are very similar inhibitors. There are differences that are key, the adding of fluorines and you know, different kinds of capping groups. But overall, the macrocycle is very, very similar, which means that all three of these inhibitors have the same susceptibility to the resistant mutants I just described for grazoprevir. So there's this macrocycle that makes them susceptible to resistance. So the trick that we're trying to do with Akbar, Ashley, and Lena is to take sort of the best parts of both of these inhibitors, ganaprevir and grazoprevir, and combine it into an inhibitor that where we pay attention to the substrate envelope and leave ourselves less susceptible to resistance. So we want to get rid of the packing against the arginine, and we want to get rid of this rigid macrocycle, but incorporate the good parts, incorporate the packing on the catalytic triad and the fit of this P1, P3 macrocycle in the envelope to make. And so we made a series of inhibitors with these characteristics and tried different functionalities to see how we could best maintain potency without the susceptibility to resistance. So here's one of the first crystal structures we solved of this new set of compounds. And even though that we didn't have the rigid macrocycle, we were very relieved to see that we retained packing against the catalytic triad of grazoprevir, and that the P1, P3 macrocycle stayed well within the substrate envelope. But we lost some potency, and so we wanted to see if we extended in the substrate envelope beyond whether we could become more potent. So Akbar, together with Lena and a new postdoc in the lab, Nagesh Rao, made a series of different inhibitors with different capping groups at the P4 capping groups or the P4, P5 capping groups. And we solved crystal structures of these to see how well they fit in the substrate envelope, and we measured the potencies of these various inhibitors against the wild type and resistant forms of the enzyme. And when we, Ashley and Chiquito solved the crystal structures of these inhibitors, we found that they did in fact fit pretty well within the substrate envelope, different variants. So these are the P4 capping, they are filling the substrate envelope pretty well, and right in here, and then the P4, P5 capping fill in additional space in here. And so then we uh, looked at the structures, and what we found was that the ones which fill the contour the protein better. So you could see that in this inhibitor, the inhibitor sort of sticks up a little bit above the surface of the enzyme, and it forms actually sort of a frustrated pocket below it. There's not enough space for a water molecule to fit down here, but the compound is also not fitting into against the surface of the protein. And compared to ones that work better, this is fitting, contouring much better within the surface of the protein. So we were interested, this turns out to be the more potent inhibitor. So when we looked at this series of inhibitors, the ones that have contoured actually fit better within the uh, onto the protein. And when we started looking at the potency, so here is grazoprevir, the Merck compound. It binds to the enzyme with about 0.2 nanomolar. When we compared it to our compounds, our best one is at about 0.8 nanomolar. Quite a few are right around 1 nanomolar. But grazoprevir loses about 250-fold in resistance against the D168A variant. And when we compared it with our inhibitors, we are losing much less fold in resistance. So we're losing more like 10 to 12-fold in resistance. So that was really reassuring that the substrate envelope is really able to help us have a flatter binding profile against resistance, and that we're able to retain pretty close to the same potency against the wild type and much better potency against resistant variants. So, I mean, this gave us reassurance that using the substrate envelope was a very powerful technique, both to define resistance, but also to give us a guide as to how to avoid resistance while retaining potency. In the last couple months, a large portion of my lab, as well as other colleagues in my department and at UMass Medical School, along with many other scientists around the world, have shifted our efforts to start targeting the SARS-CoV-2 virus. And we've been doing this in two major ways. We've been looking at the viral life cycle and working on this interface between the spike protein and the ACE2 receptor, and we're doing that in collaboration with Dr. Yang Wang at the Mass Biologics Laboratory, which is part of our university, and also targeting the proteases there with Dr. Paul Thompson's laboratory, Dr. Jaswinder Singh, and Dr. Mohsen Saeed's labs. And I'll tell you a little bit about the protease project. There are two proteases in the SARS-CoV-2. The SARS-CoV-2, like lots of other viruses, has a polyprotein with several different open readings frames. But in the first one, there are two proteases, both of them cysteine proteases that are encoded. These enzymes have already been crystallized by other laboratories, and as I mentioned, our cysteine proteases. And cysteine proteases have similar catalytic mechanisms to serine proteases, although not quite identical. Interestingly, the specificity of these enzymes is quite different than those of other enzymes. The M protease are than each other. The M protease recognizes 11 different cleavage sites within the polyprotein of the SARS-CoV-2, and with the glutamine at the P1 site. The PL protease it actually is also a deubiquitinating enzyme that targets host factors, but also cleaves 3 sites within the SARS-CoV-2 with a double glycine right before the cleavage site. So we basically have been applying our idea of substrate envelope by doing molecular modeling, and have developed substrate envelopes for both of these enzymes so as to be preemptive of resistance as we start to think about developing inhibitors. We've also done molecular modeling of inhibitors. There's been some reports on bioarchives that some of the HIV protease inhibitors that are peptidomimic also inhibit these enzymes, although very weakly. And so we've done some molecular modeling superimposing their backbones of these inhibitors onto our substrate envelope. And, looks like they're forming some nice hydrogen bonds and so forth. But initial enzymatic assays don't show that they're binding very potently. So this may not be quite accurate of how they behave, but we're trying these out. So, stay tuned. Our goal is to have be able to develop or contribute to the development of pan coronaviral protease inhibitors. We're currently expressing purifying and purifying enzymes. My colleague Paul Thompson is doing some high throughput screens, and we're setting up crystal trials to try to understand what's happening with this enzyme. So finally, I'll finish with my last story of the afternoon of trying to decipher mechanisms of drug resistance using machine learning. So when you want to decipher mechanisms, you want to have an ideal system. And for us, our ideal system is HIV. And to understand the resistance, we're bringing together two powerful techniques, molecular dynamics, parallel molecular dynamics, and machine learning to understand the mechanistic pathways by which resistance occurs. And again, just to remind you, molecular dynamics is a way for us to really understand the physical interactions that are going on inside a protein and be able to how changes in them may confer resistance. So HIV we consider an ideal model system for understanding resistance because it evolves so quickly. And just to give you a feel for how fast HIV is able to evolve, if we compare the genomic diversity that exists in a global pandemic in 1996 with the diversity of an untreated patient infected with HIV for 6 years, you see that it's about the same. And the diversity increases very significantly if you looked at the diversity of untreated individuals in the Congo in 1997. So, these are huge distances in evolution. Luckily, we have very potent inhibitors, and we have inhibitors that some of them are less than 5 picomolar in affinity. And my favorite one is a HIV protease inhibitor called darunavir that inhibits this enzyme extremely potently. When we think about resistance, we often think about mutations right in the active site, changes like this isoleucine 15 going to a valine. So where a methyl group is lost, so you have a loss of a van der Waals contact, or this 84 going to valine similarly. And because HIV protease is a homodimer, you get a double whammy. So 84 going to valine, you lose 2 methyls on either side of the active site. So understanding primary resistance seemingly is straightforward, but resistance turns out to be much more complicated than that, because in highly resistant patient variants, we can have up to 25 mutations in the 99 amino acid enzyme and still be functional. So we're interested in understanding how you can get resistance. So we took darunavir, which is this extremely potent inhibitor, as I said, about 5 picomolar in affinity. And we made a whole series of analogs where we modified the different sites on darunavir. And we saw that they all fit within the substrate envelope, and they all have actually less than 5 picomolar affinity to the wild type enzyme. And a colleague of ours, Ron Swanstrom, in his laboratory, used these inhibitors in viral passaging to select for drug resistance. So he did so over many years, and he took these 11 inhibitors and did viral passaging, where he slowly increased the concentration of drug and slowly got resistance, taking inhibitors that were able to inhibit the virus in subnanomolar concentrations and select for micromolar inhibition in very resistant forms of the virus. Now, as they did this selection. They saw a lot of changes in the sequence space. So that the sequence entropy, the variation in the sequences were very interesting. So the viral pool, when there's suboptimal amounts of drug, is able to evolve resistance, but it becomes this very diverse in sequencing. And so after the viral selection was done, deep sequencing was done at various time points during the selection, and we saw that what would happen, you'd get a big mixture of different sequences, and then a mutation would fix, and then this would happen multiple times. And interestingly, the same drug could cause different sequences of mutations to arise. Mostly it would start either with the I84V or the I50V that I just pointed out to you a moment ago. But then there would be all sorts of other mutations occurring throughout the enzyme until we got the close to a micromolar in resistance. And so on average, the number of mutations that occurred was about 10 mutations in the enzyme. And these mutations occurred all over the place, not just at a single place. So they, as I said, they usually either had the 50V or the 84V, and some had both. But we expressed and purified these different enzymes, and we saw that it actually in the protease enzyme itself, we lost a lot of affinity. And so, we were almost at the endpoint, in fact, almost had micromolar. And interestingly, the ones that most resistant variants we came up with actually had very different sequences. So there were more than one pathway to get high levels of resistance up to micromolar resistance. I'll take you briefly through the pathway of one of these enzymes, and this is a variant that has 11 mutations relative to wild type. And so just to show you, the wild type had about a 5 picomolar in affinity. Introduce one mutation in the active site, and we had about a 5-fold resistance. 2 mutations in the active site, we had 50-fold resistance. Add another 2 on the outside of the flap, and we're at 80-fold resistance. 4 more in the core of the protein. And notice these are outside of the active site, and we're up to 2,500-fold resistance. 2 more further out in the enzyme, and we're at 30,000-fold resistance. And our final one on the outside of the flap, we have 150,000-fold in resistance relative to wild type. I mean, the enzyme's somewhat compromised, but this is coming out of active viral cultures, so the virus is still viable. But what this gives us is a whole panel of different resistant variants. And so from this, we can start to discern what the molecular mechanisms are for resistance. We solved a whole series of crystal structures to try to discern this. But unfortunately, even though there were subtle differences seen in the crystal structures, they didn't really tell us what the molecular mechanisms by which the resistance occurred was. And so then we turn to molecular dynamics. And so here you can see molecular dynamics of darunavir in the wild type, and basically the inhibitor sitting happily in the active site. It's wiggling and jiggling a little bit, but not too bad. But if we look at the other extreme, the one with 11 mutations, you can see that the inhibitor is really not so happy in the active site. It starts pulling out of the active site, and you can imagine that the enzyme would completely open up if this was allowed to simulate longer. So we took the information from the dynamics, and we calculated different properties of the inhibitor enzyme complex. We looked at correlation coefficients, how correlated was the motion of the drug with the enzyme. In the wild type, it was very correlated, but with more and more resistance, that correlation fell off. We looked at the root mean squared fluctuations of the inhibitor in the active site, and with more and more resistance, the inhibitor was fluctuating more and more. And then we took all this data and we pulled it together into a machine learning algorithm. And so we took molecular dynamic simulations done in triplicate, and from those we calculated van der Waals potential, the hydrogen bonds, the torsional entropy of the different side chains. And we funneled that all into a regression analysis, an elastic network machine learning algorithm, basically led by a student in my lab, Florian Leidner. And he took not just those enzymes I just showed you, but a whole bunch of others, almost 30 different variants of HIV protease that we studied with different levels of resistance and different mutations, between 0 and 24 mutations, spanning most of the enzyme. And it pulled all this information into a protocol where we ran the simulations in triplicate, pulled out different features, like I said, van der Waals, hydrogen bonds, the fluctuations, performed a regression analysis, excuse me, where he culled the different features to make sure not to overfit the data and only use the features that were most informed the regression analysis. And then we interpolated those informative features to understand what it meant. And so by doing so, he was left with 9 features that described the data set pretty well. And he further examined all 9 of these features and all combinations of them, and ended up with a set of 4 features, which really performed a very good regression analysis. Here are the 9 different pairs that from the initial. And what was fascinating to me is that even though the HIV protease is a symmetric homodimer, this set of 9 pairs really sliced the enzyme in an asymmetric fashion through the active site. And remember that these 9 pairs were chosen out of many, many different kinds of variants that had mutations throughout the enzyme. And the regression analysis that was based on the final 4 fit the data extremely well. It not only fit the data that was used to develop the regression analysis shown on the blue circles, but also complexes, inhibitor enzyme complexes that were in our test set that were not used in the model, fit within a 1.3 kilocalories of the experimental data. And these are from variants that are way outside the active site. So other methods like free energy perturbation is not going to be able to get at this because mutations are outside the active site. When we compared this with a method that's often used, MMGBSA, this was not able to rank these inhibitors very well from 3 snapshots. So when we look in detail at these different inhibitors, these different features that came out of the regression model, we can see very specific interactions. So here's 50-84 interaction. And you can see that as these 2 residues separate during the different, in the different complexes and dynamics on average, that it correlates very closely with the experimental potency. So these are sort of bellwethers of resistance. Another interaction, which isn't that surprising, is the loss of a hydrogen bond with darunavir. I mean, this drug is making lots of interactions, but this particular hydrogen bond really correlates with the loss of affinity. More surprisingly, are remote sites that where the packing gets either more relaxed with resistance, again in a fairly linear fashion, or packs more tightly as the enzyme active site opens up and the flaps open up. These 2 residues, 12 and 13, pack closer together, again correlating very closely. So I think this method is very powerful for understanding the impact of resistance in a diverse set of different variations in a way that I don't think would be possible without molecular dynamics and machine learning together. So just to finish my talk, and I realize I'm running a little bit late, I apologize. I hope sort of quickly what the take-home lessons are is that we started out with the base editing and understanding how our innate immune system contributes to resistance, and hopefully we'll be able to decrease resistance by inhibiting ApoBex in the future. How the substrate envelope is able to avoid resistance that I showed you in hepatitis C protease, and how we're starting to initiate this work on the towards the development of pan-coronaviral inhibitors, and how hopefully with machine learning we can decipher the role of remote mutations by the role of remote mutations in conferring resistance by altering dynamics. And that through this, we are able to identify key features and mechanisms of potency. And our next steps are to incorporate this into structure-based design efforts. So I hope that it also gives you a feel for why I think in this multidimensional space of structure, dynamics, both structure of both protein and inhibitor, and the wide sequence diversity that exists in biology, and that these interactions translate to many other systems. And with that, I just want to conclude by acknowledging all the people in my group, both current and alumni, and my many different collaborators. I'm a great believer that together we are more than the sum of our parts. And with that, I'm very happy to answer any questions. Thanks so much.
Big round of applause for Professor Schiffer. Wow, thank you so much. That was just a fantastic talk and a huge overview of a multifactorial approach to dealing with drug resistance. As a drug resistance guy, I loved it.
Okay.
Does anybody out there have a question for Professor Schiffer? Especially you grad students. Okay. Well, I guess I'll lead off. You know, when you started talking about that ApoBec enzyme, and in particular how that's one of the fastest evolving genes in the human genome, that got me really interested. More so from the fact that, you know, how does it maintain its function if it's continuing to evolve at such an incredible rate? And I wanted to ask you, you know, thinking about its actual function, have they done any knockout studies for those enzymes in animal models and determined what happened? I mean, did they have increased rates of cancer or other kinds of things?
So, I mean, it's tricky to do the knockout model because, I mean, because there's such variation in the apobecs. So the mice only have one apobec. Okay, and so, you know, we can't really do knockout studies in ourselves. I mean, I don't know if there's a humanized mouse model where all 7 apobecs in AID are introduced. There may be that I'm not aware of, but it's.
You give me the impression that its effects might be lethal in mice since they only have one. Is that what you're saying, Celia? Or?
I actually don't know, so I'm.
Okay, unfair question. I apologize.
No, not an unfair question at all. I just, you know, like we always tell the students, you should say when you don't know. I don't know.
That's me for sure. Anybody else? Additional questions? I think people get shy on Zoom.
Yeah, no, and I know I'm meeting with many of you over the next day or two. So, but you know, feel free to ask now. And I know I went into the hour, so.
I did have one question, and that pertains to the study with HIV and its inhibitors.
Yes.
And you said that some of them had as many as 10 inhibitors. That was one of your colleagues' studies that was over a long term. And developed significant resistance. My question is, when they do encounter that many mutations, how does it affect the virus itself and its potency, its virility?
Yeah, no, that's a good question. I mean, when they actively select for high levels of resistance, the virus is pretty compromised. I mean, so it's not so easy to culture virus of these very highly resistant ones. But that said, there have been variants of HIV that have been sequenced from patients with high levels of resistance that will have upwards of 25 mutations in their protease gene. So even though these viruses seem very compromised, similar things have been observed in the patient population.
Okay, thank you.
Yeah.
Additional questions for Dr. Schiffer?
Yes, I have a follow-up on Trisha's question.
Yes, sure.
I'm wondering on these HIV resistant isoforms or evolved forms of the proteases. Have you guys looked at the thermodynamic stability of those enzymes?
Um, we we have looked in the past at not the stability of the enzyme per se, like by melting temperature, but we've looked at the thermodynamics of inhibitor binding, and some of them have exhibited interesting thermodynamic properties in terms of inhibitor binding uh, uh, by ITC. But when we've tried in the past to do uh, differential scanning calorimetry, um, the protease wasn't very well behaved, so it was very hard to do that. My colleague, Bob Matthews, my department chair, his lab for a while tried to study the um, unfolding of resistant variants, but the enzyme is not, once you fold it, it's okay, but it doesn't unfold and refold very readily, so it's not the easiest to do.
I guess it occurs to me that as you evolve the substrate pocket, that pocket may be evolving well beyond your models
So,
if the global structure of the enzyme starts becoming more labile and more breathable.
yeah, so we thought about that. We actually did do some investigation into that, not of the super highly resistant variants, but others. And interestingly, the substrate envelope actually was conserved as resistance was happening. So somehow that had to be maintained, presumably because HIV protease has to cleave 10 different diverse sites. So it doesn't have that much plasticity of being able to evolve the substrate envelope, recognition envelope very much. So that's sort of something that needs to be conserved.
Interesting, interesting.
Celia, I wanted to ask you a question about the HIV protease inhibitors that they actually thought would have some effect on COVID. Was this based on studies that were done with the original SARS or MERS or coronavirus, or was this something that was seen in cohort studies of people getting coronavirus infection?
I'm not, I think in the early days in China, people were trying every antiviral that they had in their coffers to try to help these people out. I don't actually know uh, or recollect whether they had previously thought that some of the HIV protease inhibitors had efficacy or not. But I mean, this paper that I cited, these drugs are against HIV are in the sub-nanomolar inhibition or single digit nanomolar. And what's seen, EC50s in the micromolar, they wouldn't be good for treatment.
Thank you.
Yeah.
Any additional questions for Celia? I got one more, just an observation. You mentioned how the HIV protease is malleable. It's incredibly malleable to mutation, yet it maintains its function. In a similar thing, in some of the stuff that I've worked on, I've been amazed by how much you can do to elongation factor G in Staph aureus in terms of mutation. You can create mutations in the entire length of the protein, but somehow the cell doesn't die. Somehow the cell remains alive and it becomes resistant, in this case, to fusidic acid, in your case, resistance to HIV protease inhibitors. Is there something about targets of antimicrobials that make them more malleable but capable of maintaining function? I mean, it seems that all proteins would be the same, but
I mean, I think that there probably is some addition, on average, additional malleability. I mean, because most virus, the evolutionary pressures are so much faster for viruses with the infidelity of their polymerases. I think that they may be a little bit more plastic than slower evolving systems like ourselves. I mean, but I don't know if anybody has done a systematic study of that, but yeah,
Thank you, Celia.
yeah, sure.
Any additional questions? All right, folks, with that, I'm going to thank our Rekel speakers.